Okay, good morning everyone. So welcome to the fifth lecture of the Introduction to Computational Molecular Biology course. So today we're going to talk about genome assembly and annotation. This is an extension of last session where we talked about read alignment to the genome. So now we're going to talk about another analysis where we take the sequencing reads and we assemble them into longer and longer contig and scaffold instead. Okay, so today's topic, we're going to talk about the concept and the algorithms behind sequence assembly. We're going to talk about the structure of genomes because once we are able to assemble the sequence and reconstruct the genomes, then we need to understand what genetic elements, what genomic elements are on the genome, such as protein coding genes or transcription factor binding site or enhancer, promoter, for example. So we're going to end today's lecture with kind of algorithms that we can use to annotate the location of genes on the genome. Okay, so when we talk about sequence assembly, the concept is very simple. Since we start with a genome, we fragment the genome when we do the sequencing because we cannot sequence the entire chromosome at once. So we kind of end up with reads that are usually very short, right? And short reads, when we have very short sequence, one of the problems is when we map it back to the genome, we sometimes are not really sure where it belongs to because there are many regions with the same sequence. So if we can kind of group the reads together and see that some of them have kind of overlapping sequence, like the diagram here, some reads might have similar sequence, and we can tell that they might kind of belong to the same continuous genomic fragment. Then we can assemble them together into contig. So contig is an assembly of sequencing reads such that there is no gap. So every contig we know from end to end what the nucleotides are. But sometimes we might have pair-end sequencing data where we know the sequence from the front, right, the forward read, and we know the sequence from the back, the reverse read, but we don't know the sequence in the middle, right? Because you might have a DNA fragment of length, let's say 1,000, right? And you sequence the first 100 nucleotides and the last 100 nucleotides in a pair-end sequencing. So you still have 800 nucleotides in the middle where you might not have sequence at all. Right? So in that case, you have kind of pair-end sequencing evidence that these two contigs should be connected to each other, but you don't know the sequence in between. In that case, we are allowed to connect these two contigs together to form what's called a scaffold. So a scaffold allows some gaps in between, but these gaps must be supported by either known structure of the genome or by pair-end sequencing data. Okay. But essentially, the goal of the assembly is to reconstruct the original genome as much as possible by starting from the sequencing reads. So this is to compare what we learned last time, like sequence alignment. We have a template, which is the reference genome, and we align each read to the template. In today's lecture, we're going to talk about assembly, which is where we try to reconstruct the genome without relying on it. And the key difference is if you have a good reference genome, like human, mouse, or other model organisms, it's usually good enough to use the genome as a reference. But if you are studying microorganisms or some other species where you might not have the exact reference genome, but you know the genomes from a related organisms, and you expect there to be some structural changes, like some chromosome might be shorter or longer, there might be some insertion, deletion, some structural variants. If you trust the reference, you're going to miss those structural variants because you are going to just align to whatever structure the genome is. But if you do sequence assembly, you have the opportunity to detect new insertion and new deletion. So it's a trade-off. assembly is much harder, but you have the possibility of discovering new things about the genome. Sequence alignment is much faster, but sometimes you cannot detect structural rearrangement or structural variants. Okay. So we're going to talk about sequence assembly for different type of sequencing data. So for short reads, this is the first thing that kind of people develop for. And it's hard because when you have short sequencing data, like, sometimes you have multiple possibilities. You have, let's say, 8 nucleotide match in the middle, like TACAAGT here, kind of match both the first 4 sequence, the middle 3 sequence, and the last 3 sequence. But if we want to assemble these reads together, we ran into a problem where before this vertical line to the left, everything agrees. But after this vertical line, there are 2 possibilities. Do you want to believe that the next sequence would be TAG or would it be CCG? Right? How would you know? Right? And this might be both true. You might have a repetitive region where one repeat kind of ends with TAG and another repeat ends with the CCG. Or you might have a polyploid genome where the DNA sequence on the father is TAG and on the mother is CCG. Right? So sometimes short read there is not enough information to distinguish between which possibility or whether all possibilities are correct. But this is what we have to work with if we analyze kind of short read data. Okay. So what can we do? Okay. First, we can try to reconstruct as much as possible, and then we have to resolve this kind of ambiguity kind of afterward. At least we want to get the first half correct, and then we have to deal with multiple possibilities later. So one way to organize sequencing data into a structure that is easy to find assembly is what's called a De Bruijn graph. So a De Bruijn graph, the names might sound intimidating, but the idea is simple. If you have multiple reads, right, these reads will be a node in the graph, and you will draw an arrow from one read to the other if these reads share an overlapping sequence. And the arrow will point from the reads that start first to the reads that follow. So these two are connected because the first read ends with a TGGAAG, and the second read start with a TGGAAG. So the suffix for the first read has to match the prefix for the second read. And if you can connect multiple reads along this graph, you can infer that these might be assembled to form a longer contigue because you see the connection from one read to the other with no ambiguity. This is a simple example where you have a linear structure where you have one read point to the next, point to the next, point to the next until the end. And you can assemble all of them into one single contigue. But most of the time, your De Bruijn graph where you construct from like Illumina sequencing data, you're gonna have branches where one read can connect to multiple reads, or you probably will have a cycle where a series of reads loops back onto itself. So this might come from like a repetitive region where you have the same reads kind of repeating over and over. So that way you have a cycle in the graph. A cycle is you can start from one and you can come back to the same position. So within this De Bruijn graph, one of the key principles of what should be the assembly that we produce from this graph is we at least want to find the path. So the path is a walk or a travel from one node to the next along the arrow. And we want this path to be very long, because if it's long, then that means we get a long contigue. Right? And long contigue is much more high confidence than short conti. The key is we don't want to visit the same node again. Otherwise, you can loop back over and over, right? And you get an infinitely long conti. So we cannot resolve the problem of repetitive region with short read. So what the best thing that we can do is we can not produce the loop. Right? So let's say I walk from AAG down to here and down here, down here, down here, down here, and I walk over into a loop. Right? I'm not allowed to go to AGGC again. Next, I need to go to AGGT and then go on to the other nodes. This way, we have a path that visits every node in this double graph. Okay. And it doesn't visit the same node twice. Okay. So this technically is called a Hamiltonian path. It's a path in this graph that visits all the nodes. So it uses all the reads basically. It uses all the sequencing data to reconstruct the conti in such a way that it doesn't reuse any read. It doesn't visit the same read twice. And it visits everywhere. So this is one of the algorithms for finding the conti from this kind of double graph representation. But most of the time, there are going to be multiple possibilities that prevent you from using all the reads. In this example, even though there are branches and there are cycles, you can still find a way to walk through every read. So there's still a Hamiltonian path. But sometimes it's impossible. For example, if you have multiple endpoints, your path has to take one branch or the other, one branch or the other, one branch or the other, then there's no way that you can use all reads. And the best heuristic, the best guess that we can make is we can remove the shorter branch because we want to use as much sequencing data as possible. So the idea of tip trimming is we're going to cut out the shorter branch and we're going to believe that the longer one is more correct. The second technique is to remove what they call a bubble. So multiple possibilities that can either come from repetitive region or come from the fact that we have polyploid genome. So we need to pick one or the other to go over this region of the genome. And you can do this by either using a pair-end sequencing data to show that one sequence has to be next to another sequence, or you can take the competition between these two sequence possibility by looking at the read count. Because notice that in this the Bruijn graph representation, we are not putting the read counts onto it. Because the read that produce AAAG, there might be multiple reads. So even though we represent one sequence as a node, each node can have more or less confidence based on the number of reads that we have with that sequence. So we can also use that to rank which part in this graph that we should travel so that we get more sequencing coverage or more read counts as we generate the content. So these techniques are what people use to try to start from the Bruijn graph and then try to cut out all the rest possible part and then end up with a non-ambiguous content at the end. Okay. So that's the idea for the short read. There are more computer science technique that goes into this, like to make this process fast and to make sure that the idea of heuristic trimming or removal is actually good. There are many things that goes inside, but for the objective of this introduction course, I think this is enough to show you the key idea behind assembly. So next we're going to think about long read data. and why we separate them from short read. So what changed when we try to assemble long read data versus short read data? The good thing is long read data usually produce less number of reads, so we'll have a smaller the Bruijn graph. And because long reads are long, when they overlap, they're going to overlap not by like 10 nucleotides, they're going to overlap by hundreds or thousands of nucleotides. So we are more confident that they are connected to each other, more than with short reads. But there are some difficulty, because we know that long read data are not as accurate. So the overlapping sequence might not be perfectly matched. There might be some mismatch. And because they are long, we can imagine that the overlap, sometimes you can have a different possibility. You might have a 1,000 nucleotide that overlaps with certain mismatch, and you might have a different read with, let's say, 800 nucleotide that overlaps with different mismatches. So how do we distinguish which one is better? For example, in this kind of quick example that I generated, so we have the first long read, and then we have two different long reads that start out similar. It overlap, the prefix kind of overlap with the first short read, the first read, sorry. But the ends look different. So which one should be connected? This situation can occur. So this leads people to think of a different assembly algorithms for this type of situation. And the overall algorithms is called a overlap layout and consensus algorithm, or OLC. And they break this algorithm down into three parts, as the name suggests. Overlap, which is the part where you calculate the overlap between two reads. This is different from the Bruijn graph, because the Bruijn graph, you treat each read as a node, and then you connect the read that overlap perfectly. For long read sequencing, you need to determine whether two reads should be called overlap or not. So, and the overlap can have different length. So the overlap steps now have to be calculated for each pair of reads with a different algorithms. And once you overlap all the reads, so you create the Bruijn graph basically for the Bruijn version for the long read data. Then you need to do the same as before. You need to trim the possibility. You need to remove the bubble to summarize the Bruijn graph into linear parts, so that you can call these into contigs. And to accommodate the low sequencing accuracy for the long read data, there is a consensus step where once you have the contig, then you can look at the long reads that map to the contig and then call the consensus sequence. So you can see that this kind of is engineered toward long read data specifically, because it allows for flexible overlapping sequence with some mismatch, and it assumes that the reads are not that accurate. So there is a call consensus sequence step as well. So let's see how it works. So we want to modify what we already know from several lectures ago about sequence alignment, because the way to figure out if two reads overlap or not is that we need to align them somehow. And we have already seen one modification of sequence alignment. When we do local alignment, we have a modification where instead of calculating the score normally, we reset our negative score to 0. And we have the argument that because for local alignment, it doesn't need to start from the front, it doesn't need to end on the back. So anywhere that you get negative score, it's better to just discard the sequence from the front or from the back. And you only find the partial match in the middle. So we see a way that we can modify the dynamic programming algorithms to fit a specific need. So how can we do the same for this problem of finding overlap? So first we have to think, what does it mean for the overlap in the context of sequence alignment? for global alignment, the alignment part must start from the top left corner and end in the top right corner. Because the top left corner designates that you are using the first sequence. You are starting from the first nucleotide of both sequences. And the bottom right corner means that you consider the last nucleotide of both sequences. When we do dynamic programming for local alignment, we argue that we don't need to start from the top left and we don't need to end on the bottom right. We can start anywhere in the middle and we can end anywhere in the middle. For finding the overlap, overlaps means the suffix or the end of the first sequence must match to the prefix or the start of the second sequence. So let's say my first sequence is the vertical one and my second sequence is the horizontal one. So that means my alignment must consist of a suffix of the first sequence, so it doesn't need to start from the top, right? It can start from the middle rows, but it has to start from the first column. Because the first column means I'm considering the start of the second sequence. This arrow, this green arrow here that starts from the first column and end on the last row correspond to the alignment between the nucleotide that they put in the boxes, which is the suffix, like the end, the last three nucleotides for the first sequence, and the first three nucleotides for the second sequence. So this is overlap between the first and the second sequence. So now we have the condition. Right? We want to find alignment that starts anywhere on the first column and ends anywhere in the last row. So how should we modify the scoring? Right? For local alignment, we say that any negative score is set to 0. In this case, we want to convey the idea that the alignment must start from the first column, but it can start anywhere. So we put 0 everywhere, because wherever it starts, there is no penalty. It can start from anywhere. And we put negative large value, like negative infinity, just for notation's sake, to designate the fact that our alignment cannot start from any columns other than the first. It cannot start from the up here, because starting from up here means that you are adding gaps to the first nucleotide of the second sequence. And you cannot do that because you have to consider the prefix for the second sequence. So this is the condition that kind of reflects the concept that the alignment that we are going to have here have to be a suffix of the first sequence and a prefix of the second sequence. Okay. So let's see how this works. So this we are going to use the same scoring criteria as before, the one that we use for global sequence alignment. So the only thing that we modify is the starting score for the first column. If you remember, it used to be -2, -4, -6, depending on the number of gaps. But now that we allow the sequence to start from anywhere, the initial score are 0. And these are all kind of minus negative value, very large negative value. So we can fill the alignment score as usual. This cell is 1 because we take A and A to be a match. This is -1 because we add a gap. Going horizontal and vertical means adding a gap. Going diagonal means having either a match or mismatch. So this is 0 because it's 1, and then -1 because G and T doesn't match. In this case, okay, the optimal part, so to find the optimal solution, we look on the bottom row here where the alignment have to end, and we find the largest number, which is 2. And then we trace back how did we arrive at this 2. So this 2 is 1 plus 1 for a match between T and T, and this 1 comes from this 0 and a match between C and C. and this 0 comes from this -1 which with a match between G and G. And this -1 comes from this score of 1 with a gap. So we can trace the alignment kind of as before, and we know that this is kind of similar to what we get from global alignment. So it might not look so different, but the principle has changed because we changed the way we initialize the score, and now the alignment that we have is an alignment that finds the overlap. Okay, so let's try a different example. So I deliberately changed the first sequence so that we find a different solution. So it's now an ATTG. And if we view the score as before, we're going to find that the best score on the bottom rows here is 1 at this position. And this one, score of 1, comes from a score of 0 and a match between G and G. And this score of 0 comes from this score -1 plus a match between T and T. And this -1 comes from a mismatch between A and T. So in this case, we have the answer that the best overlap between these two sequences involve the last 3 nucleotides of the first. Sorry, I switched the sequence 1 and sequence 2. Just notice that. So we use the last 3 nucleotides of the first sequence and the first 3 nucleotides of the second sequence. So this is an overlap, and we can create a De Bruijn graph by connecting ATTG to ATG-CT. So this is the techniques that people adapt from dynamic programming for global sequence alignment to create a De Bruijn graph for long-read data. So once we use that techniques, we can connect all the different long-reads together into a De Bruijn graph. It's going to look very messy because short read will not have so many possibilities because it's short, right? It cannot connect to too many different other reads. But for long read data, because the sequence is long, there can be multiple possibility of overlap based on the sequence alignment. So you have a lot of arrows. And many of them will look redundant. For example, if you consider the example that I put here on the top right, you have 3 reads that overlap with each other. And you also have the first and the third read also overlap because they are long enough that the overlap can contain the second read. In this case, we can simplify the De Bruijn graph by cutting all these kind of indirect connections. Because we know that we can infer the relationship from the middle reads. If these are all connected, then the first and the last is also kind of connected because the overlap kind of span the middle reads. So we can simplify this kind of a little bit. And in most of the case, if our long read data is kind of unique enough that we can determine the chromosome structure, we should end up with something like this, where we have a linear graph because this is like the only possibility for the chromosomes. And then there may be some repetitive region where there are many possibilities, many copies that can be produced, and then the rest of the chromosomes. And then we can do what we already learned from long read sequencing data. We can take all the reads that map to each linear region and then call the consensus sequence, call the majority one to get the actual genomic sequence. So this part is not new. The last possibility is when we have both short read and long read data. What should we do? The general consensus from the literature is that we believe the short read first. We generate assembly from the short read first, which might not be completely accurate. It might miss some structural variant because it's from short read. but the sequence is going to be very accurate. So the idea is we're going to assemble the short read first, and then we're going to align long read to the short read. And we're going to use the long read to correct the assembly for any structural rearrangement. If the long read suggests that there is some gap, some insertion deletion, then we can adjust the assembly. And if the long read can match to the assembly directly, then that's a good evidence that we already have the correct alignment. And when we want to align long read to the assembly, we're going to have to modify some alignment algorithms again. And people call this a semi-global alignment. And they put a semi here because there are two possibilities. The long read can match to the contig from assembled from short read directly. In this case, this is not a global alignment, right? Because it doesn't use the entire contig. But it's also not a local alignment because we want to align the entire long read. So they call it a semi-global because it's global on the long read, but not global on the contig. So you have to put a different concept so that you ensure that the alignment starts from one end and use kind of the long read data as a whole, but it can be local on the contig side. In a different possibility, you may have a long read that create a gaps in the contig, or you may have long reads that map to different contig. So these long reads can connect two contigs together. In this case, the alignment is like a local alignment, but it's not exactly local because the alignment has to start from one end of the long read, but it can terminate early on the end of the contig. So the alignment again has to be kind of customized or modified so that the dynamic programming steps knows that the solution, the answer that we want to get, has to at least use one end from the long read and one end from the contig. So we see that now that we know kind of how to modify sequence alignment, like from global alignment to local alignment to alignment for finding overlap. Now we see the possibilities, many possibilities, where we can adapt what we know about dynamic programming and apply it to different situations in DNA sequencing data. Okay. So in some case, we want to do a hybrid. Right? We know some reference genome, but we also want to do some assembly so that we can discover new structural variants. So in this case, we can mix both techniques called reference guided assembly. So basically, we're going to use the reference first. We're going to align our reads to the reference. And some of the reads will match because that's the region where the reference genome and our genome is the same. And some reads might not match. And these reads might indicate structural variation, like new insertion, new deletion, or something that differ between the reference genome and our genome of the species of the organism that we are studying. So we can distinguish the two reads, right? One that align and one that don't. Right? And for the reads that align, we can group them into blocks. By blocks we mean kind of region on the genome where these reads align to. And we can do assembly. Instead, usually when we do sequence assembly, we need to consider every read. We need to generate the bool graph from every read. But since some of the reads kind of perfectly align to the reference, and we know that their position on the reference are next to each other, we can do assembly within each block and not as a whole. Right? So the problem becomes much easier. You can create a double graph just local to these reads, and you generate one assembly. You create a new double graph for these reads that are next to each other, and then you generate another assembly. So now you have multiple contig possibilities generated from different positions on the genome and also assembly that you generated from the unaligned reads. And then you can try to combine these, kind of integrate these into a consensus contig. Like you can align them together and create a longer contig. So this way, you kind of utilize the information from the reference genome to make your assembly easier on some subset of your reads, right? And you still have the unaligned reads to suggest if there is any structural difference between the reference and the actual genome being studied. And this technique is very important if you study, let's say, virus and bacteria, which kind of evolve very rapidly. So their structure, the structure of their genomes can change a lot, and you are not going to have the most recent reference genome for every species. Like you might have a reference genome, like from a different strain that people studied 5 years ago, like, and you isolate a new strain today, and you want to reconstruct the new strain's genome. You can use the data from 5 years ago as a reference, but you also want to consider the possibility that during these past 5 years, maybe the bacteria have accumulated like new genes or lose some genes. So you want to combine both past reference genome with the ability to do assembly yourself. So this is a kind of hybrid technique. So now that we have seen several ways to do sequence alignment, we need to know how to evaluate. Because sequence assembly. Because when we do sequence assembly, we essentially have no answer. Whatever we assemble is new. So how can we tell if what we are getting is good or not? A simple metric is the number of contigs. Because in a perfect world, the number of contigs, which is the number of consecutive kind of continuous sequence that you can reconstruct, like in an ideal world, that number should be equal to the number of chromosomes, because that's the unit of continuous DNA that you can construct. You can never map across chromosomes because they are not the same molecules. So the contig count can tell you how close you are to the number of chromosomes. If your assembly is very fragmented, like you have a lot of short contigs, like 100 contigs, and you know that there's only 20 chromosomes, then you know that the assembly, the sequence might be accurate, but the completeness in terms of the structure is not so good, because you have 100 fragments that should represent only 20 chromosomes. So that only consider like the numbers. The next metric, called N50, is the length of the shortest contig that form at least 50% of the total contig length. So if you imagine that all your contig that you get together reconstruct the whole genome. So this N50 asks, okay, if I only consider half of my genome, how short of a contig that I need to consider? Let's see the number first. So let's say we have 5 contigs with length 2, 3, 5, 7, 9. So 50% of the total length would be 30. So if I want to select contig from long to short that covers at least 50% of the length, I need to select 7 and 9, which together give me 60, greater than 30. So in this case, my N50, the length of the shortest contig that form at least 50% of the total length would be 7, because I only need these 2 contigs to form at least 50%, and the shorter one is 7. So this number tells you if this number is low, like 1,000 base pair, then that means in order to cover 50% of your genome, you have to rely all the way to short contigs that are 1,000 base pair long. And that sounds very bad. If you have to believe contigs that are as short as 1,000 base pair to cover 50% of your genome, then you are not going to have a good reconstruction of the genome. But if your N50 is high, let's say 1 megabase, that sounds very, very good, because you can recover 50% of your genome by trusting contigs that are 1 megabase or longer. So this gives you another sense of the quality or the completeness of your assembly. People sometimes also suggest a new matrix called U50, U from unique. So you can think about N50 as the length of the shortest contig that kind of form the total of contig length. But some people argue that if your sample contains more than one species, let's say host and pathogen, or if your sample, like from bacteria, have plasmid, or if your samples come from environment, like water, and there are background DNA, some of your contig is not going to correspond to the genome, or at least not to the genome of the species that you are studying. So they think that by using N50 which consider all contig, it's not a good estimate, because you essentially want to assess the quality of the assembly for a particular genome. So for U50, what they suggest is you should align, or if there is a way for you to tell which contig correspond to the genome of the organism that you are focused in, you should only consider those contigs and not every contig. Sometimes that's possible. You might look at like a GC content and identify which GC content can match your organisms. Sometimes you know the host, so you can align all your contig and then remove anything that align to human, so that all the rest will be your pathogen. But sometimes it's not very possible. But still, this is just to give you ideas of what is possible. Okay. Another way to check the completeness of your assembly is to check whether your assembly contains all the core genes or all the important genes in that organism. And one of the popular tools is called BUSCO. So the idea of BUSCO is, now that we have information from the genome for many species, we can create a database. And in this database, we're going to compile for each taxonomic group, like in plants, in animal, in mammal, in bacteria, in virus, which genes are present consistently. So if you reconstruct a genome from that species in a particular group, you are bound to find this gene. So BUSCO kind of takes this approach to study all we know about genes, groups them into clusters, and when you have a new sequence assembly, you can submit your assembly to BUSCO and specify the taxonomic group, and they will tell you whether your assembly contains all the genes that should be found in those taxonomic groups. So let's see what their result looks like. So let's say you put in 5 samples. They're going to give you some of these keywords: complete, fragmented, missing, so CFM, like complete, fragmented, missing. And for complete, they're going to tell you if it's single copy or duplicated. Okay, so let's interpret what this means. So complete means that in your assembly, they can identify the core gene as a complete gene, like from the start codon to the stop codon. If it's fragmented, then they might only find the first part of the gene or the latter part of the gene, but not the entire gene. If it's missing, then there is no trace of that gene in your assembly. So this can happen when you have a fragmented gene. That can be because your sequence coverage cut in the middle of the gene. So you only have the first half or the second half, but not the entire gene. Or you might have a incorrect assembly where instead of assemble the entire gene, you connect the read from the middle of the gene to somewhere else. So you create a different kind of incorrect genome. If it's missing, then you might not have any sequence coverage in that region. So you might need to sequence more. Okay, when it's complete, that means it's good news. Your assembly contains the entire gene. You can find single copy, so only one copy of the gene in the entire assembly, or you might find multiple copies of the gene in the assembly. In the case of Busco, the genes that they select to test are the genes that they expect to be only single copy, or at least they are found in single copies in other species in the same taxonomic group. So if these are found as duplicated in your data, it could suggest an assembly error where you might accidentally kind of assemble the same region twice. So you might have sequencing error, or you might have some other errors that makes the assembly algorithm think that there are two different regions for this gene. So let's say for sample 1, we found 750 genes in a complete structure, only 9 fragmented, and missing 200 of them. So this doesn't sound so good. We are missing like a quarter of core genes that should be found. And among completed genes, most of them are single copy as expected. But still, we have like a 5% of them that's duplicated. So we kind of might need to inspect our data a little bit more. For sample 3, this looks very good. There's only 148 genes that are related probably to this taxonomic group, but most of them is found as complete. No fragmented, only 9 missing. And all that are found as complete are single copy. So this is a good sign that your assembly might be very near complete. But again, because Busco only focuses on core genes that are found in single copies, and this gene might be located on certain position on the genome. So this doesn't mean that you have good assembly for the rest of your genome. It's only an approximation that if your assembly does well on this gene, it probably does well overall. If you assume that the assembly error is not biased to a certain position. Okay. So with Busco technique, we realize that we need to be able to at least kind of annotate genes on our assembly in order to assess the quality. So this would bring me to the next topic, where we want to talk about the structure of the genome and the kind of genomic elements on the genome, and how we can annotate them in our sequence data. So we're going to rely only on sequence to make these predictions. Okay. So let's think about the structure for the genome. In simple organisms like microorganisms, most of their genomes are genes. And this graph shows very well that if you increase the genome size for microbes, the number of genes scale in a linear fashion. So for a microbe that gains more genome, it does so because it's gained more genes. The genome becomes longer because it's gained more genes. But when we come to higher order organisms like eukaryotes, the situation changes. Many of you probably have seen this distribution before. This is a component of a human genome. And genes only consist 1.5% of the entire genome. I mean, we doesn't take into account the non-coding genes here, but still, it's very small. And the rest are like repetitive elements, introns, non-coding stuff, duplication, some weird insertion from other microorganisms. So it's very complex. And this graph, this table, sorry, shows you that if eukaryotes expand the genome just to accommodate genes, like in bacteria, humans should have 3 million genes. But instead, we only have 20,000 genes. So we are using the bulk of the genome for something else, and not for genes. And this holds true for every eukaryote. Our genomes is no longer about protein coding. It's about many different elements, many different regulatory mechanisms. But let's focus on the simple structure first. Let's focus on the gene structure. We know that for a gene, we're going to have a promoter. We have the open reading frames that start from the start codon and stop at the stop codon. We have exon intron structure because we have RNA splicing. And we have the transcription termination sequence. We have the distal control element like enhancer or silencer that can control the transcription of the gene. So even if we consider just a gene, there's still many smaller elements within the gene that we want to be able to annotate. Like we want to say from this base pair to this base pair is the promoter. From this base pair to this base pair is my first exon. From this base pair to this base pair is my intron. We want that level of annotation. So just to expand, like we saw the example of the structure for the gene and regulatory elements, but people also try to do annotation for other things like non-coding RNA or other repeats as well. And I want to remind you before we go into the algorithm section that these annotations are best guided by data from other experiments. Like if you do RNA sequencing, you don't need to guess the start or the end or the position for the intron because you get all those from RNA sequencing. When you map RNA read to the genome, you get your read is going to map to exons. So that can tell you exactly where things are. And if you perform SHIP-seq, you're going to know where transcription factor binds, so you know the position for certain regulatory element directly without having to guess from the sequence data. But for the sake of the computational side of this course, in the next algorithm that we're going to talk about, we're going to rely on just the sequence data. But as a biologist, you should think about non-computational support as well. Okay, so what can we do to annotate protein coding gene? We need to think about the general structure for the protein coding gene. What does it look like on the genome? Let's say we encounter a start site, like AUG. If it's a single exon gene, like very short gene, then we found the stop, and then we encounter intergenic region, and then we repeat to find the next start site, and then the next exon, and then the next stop, and then intergenic region. But if our genes are more complicated, like in eukaryote, we have first exon, and then we have to identify where is our first splice site, the first intron, the first splice acceptor site, the next exon, and the next donor site, the next intron, the next acceptor site, the next exon, like in the loops, over and over, until we found what we think is the last exon, which contains the stop site, and then we go to intergenic region. So this diagram kind of gives us the complete cycles of the states that we will encounter if we were to find a gene. The problem is, how can we map DNA position from our reads, from our assembly, onto this diagram? we're going to use what's called a hidden Markov model. But before we go to hidden Markov model, I want to introduce the non-hidden version first, even though I'm hiding something here. So a Markov model is a model of states, because when we want to annotate these genomic structure, we're thinking about them as states. Start state, exon state, intron state, S state. So Markov model is a probability model of states that can change over time. In the case of sequencing, time is the position in the genome. We can think of going from 5' to 3' as going to time. And when we encounter a gene, the state change from intergenic region to start site to exon to intron and then back to intergenic region. So we can kind of use the model that describes time and use that to capture model that describe position on the DNA sequence. And this is called a Markov model. So this is a keyword that if you are interested, you can study a lot more about. Okay. So let's see this example. So we have two possible states, and this is a model for a weather forecast. So let's say from the start, like the first nucleotide or the first day, like in our life, like we have no idea what the weather will be, but we might have some prior knowledge that if we live in a tropical area, like we have a long rainy season, we might expect like 60% of the year to be rainy and 40% of the year to be sunny. So to get to the first state, like we use some prior knowledge that we expect 60% of the time to be rainy. But once we enter the rainy state, whether the next day, whether tomorrow will be sunny or will be rainy, depends on what today is. Like, because today's weather can be used to predict tomorrow's weather. So in this diagram, if today is rainy, there's 70% chance that tomorrow you're going to get more rain, and 30% chance that the rain will end. If today is sunny, there's a 60% chance that tomorrow will remain sunny, and 40% chance that tomorrow you'll get rain. Notice how these probability sum to 1. 0.6 and 0.4 sum to 1, 0.7 and 0.3 sums to 1, 0.6 and 0.4 sums to 1. So the arrows that point out from each state, either to itself or to the other state, has to sum to the total probability, which is 1. And in this case, there's nothing hidden. This is not a hidden model. What we observe from the data is the state. For the first day is sunny, the second day is rainy, third day is rainy, fourth day is sunny. So we know the weather. But in DNA sequencing data, when we want to annotate the gene structure, for example, we are not going to be able to observe these states. Instead, we are going to see the nucleotide sequence, which are not these states. So we need to rely on a hidden Markov model. So the hidden Markov model is an extension of Markov model where, so the top part of the Markov model, you have all the states, right? And you have all the transition probability, right? What's the probability of getting the next state given the first state? Right? But with a hidden Markov model, we are assuming that what we can observe are not the states, but the outcome from the state. In the case of this kind of weather model, we might not be able to observe the weather directly. Let's say you live in a different country, but you're following someone's blog. Let's say you live in Thailand, but you are following some K-pop idol, and you look at his or her Instagram every day, and you see that he or she is taking a walk today, is going shopping today, or is cleaning his or her apartment today. So you already have access to the activity for that idol. But what you want to infer, you want to predict the weather in South Korea. So this becomes a hidden Markov model because the actual weather in South Korea is hidden from you. But that's what you want. So the observation, which depend on the hidden state, are hidden from you. And you can only observe the activity for that person, whether it's taking a walk, going shopping, or doing cleaning. But what you have, or what you can reconstruct from past knowledge about the weather in South Korea or the activity of people in Korea, is that you might be able to estimate that if it's rainy, I hide, but the state on the left is rainy, the state on the right is sunny. If it's rainy, then there's only 10% chance that people will walk because who wants to walk in the rain. And 40% that people will go shopping because it's still indoor. And 50% that people will do cleaning because they don't leave their home, don't leave their house. If it's sunny, then people want to go out. 60% will go to walk. 30% will go shopping, like they still commute to shop, but only 10% stays home to do the cleaning on such good days. So we have some information that link what we can observe to the state, but we don't observe the state directly. So how can we decode, or how can we predict the state from such model? So the idea is we are given the observation, in this case walk, shop, clean. What is the most likely sequence of the hidden state, like rainy or sunny, for these 6 consecutive days in South Korea? So let's consider a small example. We are not going to do the entire 6 time point decoding. We're just going to consider the first 2 time points, the first 2 observations, walking and walking. We have 4 possible combination of the hidden state. Right? Whether it's raining on both days, raining on the first day and then sunny on the second, sunny on the first day and rainy on the second, or sunny on both days. We haven't have a statistics kind of recap lecture yet in this course, but I think conditional probability is something that you are familiar about. So conditional probability says that if you have 2 events of interest, like, and one depends on the other, probability that you will observe A, given that you already observe B, is equal to probability that you observe both A and B with no particular condition. Both of them are observed, divided by probability that you observe B. And this is just the definition of conditional probability. So what we want to compare here is these 4 conditional probability. Because we already observed our South Korean idol taking a walk 2 days in a row. We want to know if the probability that is rainy on both days, given that we already observed walking and walking, whether it's higher than the other 3, or if it's something. If we write this conditional probability out, it will be the joy probability, like observing rainy and walking together, divided by probability of walking 2 days in a row. And because we notice that the division by probability of walking and walking is the same in all 4 terms, like if we want to compare which one is greater, then we can just calculate the joy probability. We don't need to care about the division, the divisor. So we can just calculate the joy probabilities, and which one is higher, then we think that one is good. So let's do that. So some of you that might not be very well versed in probability, when we see situation like this, where the probability that we want to calculate come from actual phenomena, the best way to think about this to think about the sequence, like the steps. of that process, how it happened. In this case, because it's a time, like data over 2 days, so the first day must happen first. The first day, whether it's rain or it's sunny, that must happen first. And then the people, like your South Korean idol, who were affected by that weather situation, makes the activity. So the observation one that you get is given the weather state for the first day. And then we come to the next day. The weather for the next day depends on the weather from the first day. It doesn't depend from your idol's activity. He or she cannot command rain. So the weather state for the second day only depends on the weather state from the first day. And in this case, we're going to assume that the activity, the observation on the second day depends only on the weather for the second day. In this case, we're going to simplify a little bit, and we're not going to assume that if you already take a walk, then the next day you probably don't want to take another walk because you are getting bored. So in this case, we are going to assume that the observation only depends on the state. So we have these 4 steps in our phenomena. Right? The weather from the first day, the first activity, the change in weather, and then the next activity. So we can break down the joy probability. I'm taking the example for the rainy and then sunny as first the probability of rainy on the first day, then the probability of taking a walk given that it's rainy, then the probability that the next day will be sunny given that today is rainy, and then the probability that he or she will take a walk given that the second day is sunny. And each of these we can read from the diagram. The probability that it's rainy from the beginning is 0.6. The probability that someone will take a walk given that it's rainy is 0.1. The probability that tomorrow will be sunny given that today is rainy is 0.3. And the probability that someone will take a walk given that it's sunny outside is 0.6. So we just look up the number from the Hidden Markov model. And then we can calculate the probability for this possibility, rainy and then sunny. So we can do this for all 4 of them. And we can notice, without having to actually calculate anything else, that the last one is probably the best. 0.4 times 0.6 times 0.6 times 0.6. And that's because the other one, you can notice that if there is a rainy day, there is a very low chance that someone is going to take a walk. So this 0.1 can tell us quickly, without having to calculate, that these are not going to produce the best possibility, the best kind of prediction. And this kind of, for this data in particular, you can use intuition that if we look at walking and we see that it's only 0.1 if it's rainy, it's 0.6 if it's sunny. So if you see someone walk twice in a row, because the probability of walking given sunny is much higher than the probability of walking given rainy, it's probably, both of days are probably sunny. Even though the probability of having sunny day is lower than probability of having a rainy day in general, if you don't have any information and someone asks, is it rainy or sunny? You're going to answer rainy because it's 0.6 versus 0.4. But now that you see someone taking a walk twice in a row, even if sunny is less likely overall, it's much more likely now, given that you know that people take a walk. So this is the magic of Hidden Markov model. You can make good inference about the state, even though you are not observing the state directly, because you observe the activities or any result from the state that can reflect the state very well. So this is how we kind of decode the Hidden Markov model by hand. But if any of you want to study further, I'm not going to explain this in this class because it's too complex, but
there is an algorithm called Viterbi algorithms, which is a variation of dynamic programming. So that this term comes up again and again. And this is because if we want to reconstruct the hidden state for our observation, like let's say we have 6 observations, the best hidden state reconstruction when you arrive at the last observation is going to depend on what is the best reconstruction that you can make for the first 5 states. So in a sense, it's a dynamic programming because your solution with n observation is based on your solution for n-1 observation. So that's why this dynamic programming approach kind of comes up again and again in bioinformatics. Okay, but cool. But anyway, just want to let you know that anytime that we want to solve a hidden Markov model, we're not going to do it by hand. We have a dynamic programming approach for it. Okay. So I think we are familiar enough with hidden Markov model with a weighted example. So let's think about the model in terms of biological data. So let's say we want to do a hidden Markov model for a CPG island annotation. We want to annotate whether a certain part of the sequence is a CPG island or not. So we might create like a very simple model where we have only 2 states, whether it's a CG-rich region or if it's a normal region. And let's say we have no clue which proportion of the genome is CPG island. So we assume that it's both 0.5. 50% you can either encounter CPG island or you can enter a non-CPG island. In a CPG island, because it's CG rich, you're going to assign more probability to C and G. In the normal region, let's say you don't know anything about the nucleotide frequency on your genome, you can use a quarter each. But if you know the GC content of your genome, then you might adjust this accordingly. And then you might assume that, okay, if I encounter a CPG island, if I think I encounter a CPG island, it's not going to be very small. Right? CPG island is a long stretch of C and G. So you probably want to stay in the same state. In this case, with probability of 2/3, 0.63, you want to stay as CPG island. Only 1/3 that you want to goes back to being a normal non-CPG region. And if you are in a normal non-CPG region, you also probably will stay in the same region with more probability. So this kind of simple models kind of capture some basic notions of CPG or non-CPG. Not sure if it's a good representation or not, but this is a good start. Right? And you can adjust like the probability, so you can tune the nucleotide frequency based on how much you know about this genome. So this is still a simple hidden Markov model because you will have 2 states, right? And the observation is only 1 nucleotide. But we can imagine that if you want to annotate genes, right, seeing 1 nucleotide is obviously not enough. How can you identify a start codon with 1 nucleotide? Right? You need to see A, T, G in order to tell that it's a start codon. So there needs to be some modification, which is going to be too complex for this class, but I'm going to just mention that for annotating genetics element that require multiple nucleotides, the hidden Markov model can be modified so that each state not only produce a single nucleotide, it can produce a certain fixed length nucleotide or variable length nucleotide. For example, the end of an intron might have a certain pattern. The start of an intron might have a certain pattern, like this, because the splice donor and splice acceptor have different nucleotide pattern. So if you identify sequence GTAAGA, it fits intron start more than intron end. So if your hidden Markov model, when you reconstruct the state and you want to calculate the probability, if you are assigning intron n to this state, it's going to get less probability than if you assign this as intron start because of the pattern GTAAGA. And notice that if you assign this as intron n, because of the structure of the gene, you are forcing the next part to be an exon. Because you just end an intron, so you need to start a new exon. If you assign this as an intron start, you are essentially forcing the next part to be the body of the intron, and the part before that to be an exon. So notice how in Hidden Markov model, you are not making inference only at each position. Decision made at each position affect the states before and after, which also have to match the DNA sequence occurring before and after this pattern. So as a whole of the assembly of the genome, this Hidden Markov model, if you find the explanation that can fit each of these pieces very well, it's likely that it's going to give you a correct annotation of the gene structure. Because it has to fit many longs and key. It has to fit all the exon position. The length must be correct, like divisible by 3. The intron start and intron end has a particular pattern. So these together constrain the solution from the Hidden Markov model toward the correct answer. And the only way we can make sure that our Hidden Markov model can interpret the probability of that an exon will produce this nucleotide or the intron will produce this nucleotide, the only way we can do that is this model has to be trained. The number of the probability here, that we say 0.3, 0.4, 0.1 here, it has to come from some evidence. We are not just guessing. In the case of CpG, we also can adjust these numbers if we know something about the genome. The frequency of the nucleotide, or the length of the CpG island on average, or the frequency of A and T that can occur in the CpG island. This knowledge allows us to adjust all these probabilities. So the same thing, if we have data from related species, like we know the gene sequence from related organisms in the same taxonomic groups, we know the pattern for their exon and intron, we can use those information to optimize the probabilities for our Hidden Markov model for the organism that we are studying. So when you do the gene prediction these days, they are not going to ask for just the assembly from you. They're going to ask you which taxonomic groups your organism belongs to, so that they will shift their Hidden Markov model to the one where the probabilities are optimized for that taxonomic groups. So that's everything that I want to cover for this lecture. See you next time.